Begins. This is August 5th, 2025, Summer Bio 105. This is our study session for the lab final, okay? So basically, I don't even need the projector, okay? The lab final's tomorrow. We're going to go over what is included, the broad topics on the lab final, and then I'm going to sort of open it up where you can ask me specific questions about anything on the lab final, okay? We're done when you tell me we're done. I'm getting paid for the full 3 hours, so I'm here. You know what I mean? I've also got your old exams and your old quizzes if you want to look over them, all right? So, you know, you can do that too if you'd like. But I think everybody should be prepared with the materials that are going to be on the exam. So let's do that. Then I can pass these out if anybody wants them. And then we'll do question and answer session. All right? I've got a copy of the lab final right here. I'm just going to do what we did last time, where I make notes. Cool? Sick. Okay. Conversion between units. You know how this is. All right? Milligrams, micrograms, nanograms, right? All that stuff. Be ready to convert between the units, just like on the first midterm. Okay. Given initial and desired concentrations, be able to figure out necessary dilutions. I hate trying to spell necessary in front of the class. It happens a lot. That might be wrong. Okay. I did it?
I heard somebody had a mnemonic that was one color, two sleeves.
That's really good. That's really good. One collar, two sleeves, because of C and the S. Thank you. I'm going to use that and then pretend that I made it up and give you no credit, because that's what we do. It's just to get you prepared for academia. You know what I mean? Yeah. Awesome. So, hey, the initial concentration was 2,000 micrograms per milliliter. What dilution would you have to do to make something that was 20 micrograms per milliliter, right? And it's a 1/100 dilution. Pretty straightforward sort of question right there. Fair enough? Okay. You need to understand A260 to A280 ratios and the equation. Do I have a question about the equation? Yeah. 1 A260 equals 50 micrograms per milliliter double-stranded DNA. All right, remember these from our DNA discussion of how you can use absorbance out of the nanodrop to determine purity of the sample, as well as concentration of DNA within the sample. Solid? Okay.
Wait, what did it say before DNA?
Double-stranded. That's DS. Yeah. And that equation is in the protocol that goes over quantitation of DNA. Yep. So you don't need to have it memorized or anything like that. It's right there in your protocol, which should be in your lab notebook, right? Okay. Ugh. Yeah. I'm going to just put that. Where's my chalk go? I'm going to put dilutions with 3 exclamation points. Exclamation points. Explanation points, John. 3 exclamation points after it. All right? Because I've got dilution stuff coming out the ears here. including things like C1V1 equals C2V2, serial dilutions, C sub I times D equals C sub F. All right, all of the stuff from that chemistry review worksheet. In fact, the last page of the lab final is just dilutions and solution making. And it is 4 questions that are 3 points each out of 100. There's another question here that's got 1, 2, 3, 4 parts, 2 points each. So that's 8 points right there. So that's 20%, all right, of the, 20% of the final. Exclamation point, all right? So how would you make this solution, starting with this solution? Or what if you took this much of initial solution and added it to this much water? What would the concentration be now? What dilution did you perform? Things like that. All right? Cool? There's the question straight from the first lab midterm of, if you wanted to make 500 milliliters of 1x TAE buffer, then you should add blank of 50x TAE buffer to blank of water, right? You remember the questions like that? That's 20% of your of your lab final. I really think dilutions and solution making is an important thing that you should know leaving this course. And if you haven't taken chem already, you're going to go in there and already be able to knock that stuff out of the park, which is nice. Okay. Enzymes. I've got how many enzyme questions here? Looks like 1. I know I've got some multiple choice. 2. Okay. I've got 1 short answer and 1, 2 multiple choice enzyme questions. All right? So you need to know enzymes, substrates, all right, delta Gs, cofactors, all the stuff in the intro to your polyphenol oxidase lab. All right, the potato one. Cool. Chromatography. Chromatography, especially calculating RF values. The retention factors, right? Question.
Do you know anything about inhibitors for the enzymes?
Are you asking me if I'm going to reuse the question on lab midterm 1?
Yes.
Yeah. So, you know, my other way of writing this would be like ways of inhibiting enzyme activity. But remember, folks, every single question can be answered from the protocol. That's a standard challenge. Every time I've taught this course, I'm like, if you can show me any question on the lab final that doesn't have an answer within the protocols, I'll give you the credit for it. Nobody's ever gotten points out of me because I'm very careful that everything can be answered from the protocols. All right? Keep it in mind, but don't use it as a way to, not prepare. If anything, just prepare a really nice lab manual with lots of easy check, little tabs and stuff. All right, ways of inhibiting enzyme activity. Okay, chromatography, especially calculating retention factors. Yeah. I'm going to put and predicting movement based on solubility, if that makes sense. So remember, we're separating things based on their solubility. If we're using a nonpolar solvent, nonpolar molecules move the farthest. Solid? Cool. Ooh, and I'm kind of a sneaky snake on the way I edited that question. So be prepared. You can make, if you're filling out notes on this right now, this is something that you'd put into your protocol, you know what I mean? Or your lab notebook, of course. You could write, John's going to be a sneaky snake on this question. All right? So you pay more attention to it. That's free information. Okay. Absorbance spectra. And color. All right? So remember, green pigments don't absorb green wavelengths of light. Cool? I'm going to need the board again. So, this is sort of your opportunity to take a picture, or you can have me slow down and I'll, let you write the stuff down. Whatever. Really, it's your study session. Cool. Everybody got all this? Need me to clarify something that's written that maybe my handwriting's not legible or something? Nice. Very cool.
Which question did you say was going to be the tricky one?
I said I would be a sneaky snake, specifically, on this question right here. So predicting movement based on solubility. I try to be tricky with it. Yeah. Okay, we all good? Let's erase some text. Very exciting. So we're currently at question 7, though one of the previous questions had an A, B, C, and a D part. And there's 22, so we're making good progress through all the topics. Essentially, it covers everything on everything that we've talked about in the entire course, except for plasmid maps. Remember, I'm not going to ask you any plasmid map questions. All right? Basically because we covered it yesterday, and I don't expect you to really memorize any of that. And that's okay. Next topic. Standard curves. All right? Cool. Use the equation. And dilute things to fit on them. What do I mean there? I mean if you've got a standard curve that runs from 100 to 1,000 concentration units, all right? Let's call it micrograms per milliliter, because why not? Then you can only trust a sample you can only trust predictions of concentration between those values, right? That's why we had to dilute the soda, because it was way over here, and we couldn't trust any estimates of glucose concentrations that were outside of our standard curve. We can only make predictions within that range. We cool with that? Solid? And don't forget, right, diluting things to fit on them, and calculating concentration before dilution. Because you can calculate the concentration of the diluted sample, but we're not usually really curious about the concentration in the diluted sample. We're curious about the concentration prior to dilution. Like with the soda, when we dilute the sample to fit on the standard curve, and then we calculated how much sugar was in the soda before the dilution. Does that make sense? Okay. I love it. That got us through 2 whole pages. Once you get to question 9, there's like 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. This whole range is really very mellow, all the way up through question 19. Can be done pretty quickly. We've already covered that. We've already covered that. We've covered this with enzymes. Okay. Gel electrophoresis. All right. Yeah. Let's say Y bands separate. And ladders. Remember what the DNA ladder is, what it's meant for. Okay. Boom, 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 boom. PCR. Let's go with steps, components. Steps, components. Yeah. I think that's kind of vague, but good enough for an open note final, you know? You need to know the purpose, what components go in the PCR reaction, what each one's purpose is, what the steps are for the PCR, what happens in each round, right? Standard issue stuff. Cool. That gets us to question 20 out of 25 questions. Okay. So we're almost through. Next one, actually, the next 2 questions are Hardy-Weinberg. All right. So you need to be able to determine allele, frequencies, genotype, frequencies, yeah. And calculate a chi-squared value. And interpret it. I give you a chi-squared table when you need it, just a little one. That brings us to questions 22 through 25. Questions 22 through 25 are all about dilutions and solution making. Everything can be completed based on the chemistry review lab handout. It has instructions on how to do all the calculations you need to do. But yeah, and I'll make an announcement during the actual lab final. Like, don't forget the back page, because I've had students who didn't finish it and it's like heartbreaking. It's only 12 points, but you know, they could have done it. I know they could have done it. So, 25 questions, but many of them are short, right? The ones that'll probably be the most time consuming are the Hardy-Weinberg questions, okay? But if you want practice on Hardy-Weinberg, do the worksheet again, all right? Do the Newt worksheet. Oh, that'll be the tip. For practice on a Hardy-Weinberg, you can just rock that Newt problem over and over again. Okay. So that's everything that is on the lab final. That's it. All right. If you would like to look over your lecture midterm or your lab midterm, I'm now going to pause the recording for a moment and let people get those, okay, so they can look at them. And then we'll do Q&A. But I'm just going to pause the recording for a moment. All right, recording resumes. This is now the question and answer period. So those of you in the room, you've made notes of what's going to be on the lab final, and you've got the lab midterm in front of you. Are there any questions or any specific topics? Yes?
Sorry, I didn't mean to cut you off.
No. I was just going to keep talking forever and say nothing new. So you cut me off at a perfect point. But I would like to say, no, go ahead, please.
Electrophoresis, the bands separate because of the size of the DNA molecule?
You got it. And excuse me, I don't know what's going on with my throat. So here's a well, right, where the DNA sample gets loaded. You run the gel and you get these bands, all right? Longer sequences of DNA don't go as far. Shorter sequences go farther. Remember, this is where the negative charged end is. This is where the positive charged end is, because the DNA molecule has a net negative charge. So it gets pulled through the gel, which is basically just a physical barrier that slows movement, and bigger molecules get slowed more, so they don't go as far.
And then if they're homozygous, that specific band will be thicker than the heterozygous, or
It's
not always?
Not always. It's more that if you were looking at a homozygous individual for, and this is, homozygous, heterozygous, sometimes the two alleles can be the same length. This is too in-depth for what you need to know for your lab final. Your question's not even going to be specifically on ALU inserts, so I would say don't worry really about that too much. The banding pattern can sometimes indicate if you're looking at a heterozygote would have 2 bands, a homozygote would have 1 band. Yep, not necessarily darker, but oftentimes it will be, because there's more DNA product generated. Anyway, solid?
Yeah.
Okay, great. You had a question right there, yeah?
I was going to ask, I'm sure you can find it in the protocol.
Everything can be found in the protocol, but we can talk about it here.
The purpose of the ladder?
The purpose of the ladder is a standard, right? So if you've got the ladder loaded here, it's got a bunch of fragments of known size in it, and it'll end up, you run it, and it just has a bunch of bands that it produces. And those bands will be of like a known length. This band is 1,000 base pairs. This is 500. This is 250. This is 125. This is a 50 base pair band. And then you can determine, oh, this is a 125 base pair piece of DNA, and this is a 500 base pair piece of DNA. Yeah, cool. It's like running a standard curve, You know the distance that the fragment would travel if it was a certain size. I'm not going to go there. You could generate an equation out of this, like distance traveled based on size, and then plug y equals mx plus b into it. But we don't do that. We just run the gel with a ladder in it.
And then am I confusing 2 different labs when it comes to like the length? Let's say it's the 125. Is that? I think I'm confusing it with the allele frequency image. But does it have anything to do with like the type of what's being expressed by the DNA itself, the length it goes?
No. Yeah, I mean, yes, because it's all connected, right? You might be thinking about the presence versus absence of the ALU inserts. That's what you're thinking about? Yeah, totally. So if you had a heterozygote that had 1 chromosome that had the ALU insert, then that amplified region would be longer. And they would have another, if they're heterozygous, right? And the other chromosome would have a shorter fragment because it doesn't have the ALU insert, and it would make a band that traveled further. Okay, but just to reiterate, the questions on your lab final are not specific to ALU inserts. They are general questions about gel electrophoresis, because that's really what I want your takeaway on that topic to be, is like, what is gel electrophoresis and what can it do? And the main thing it can do is separate amplified regions of DNA based on their length. Cool. Additional topics that you'd like us to, yes, please.
I'm wondering if it's worthwhile going over turgidity again. It's another one of those things. I'm curious to.
Well, you know, I didn't write it on the board. And I wrote everything on the board that's on the lab final. And while I love talking about osmosis and turgidity, and osmosis is incredibly important, I think that I'm going to just say, we can talk about that not during the lab final review session. Even though it's seriously, it's one of my favorite things to talk about. I love talking about osmosis. This is difficult for me right now to say that let's not talk about that. But we'll focus only on things that are on the lab final here. Any other topics that we can review? Yes, please.
Can I ask you to go over like a specific question from the
Yeah,
midterm?
absolutely.
I was gonna ask about number 3 with the it's prepare 150 milliliters of a 2-molar solution of NaCl.
Okay, solid. Prepare 150 milliliters of a 2-molar solution NaCl. And we knew from the previous question that NaCl has a molar mass, right, or molecular weight of 58.44 grams per mole. Correct? Is that correct?
Yeah.
Sick. All right, so here's how we set that up. What do we want to know? We want to know, this is one way of doing this, grams sodium chloride, all right, per every 150 milliliters. Okay? So if we know what units we want to have, right, then we can set up the dimensional analysis to give us an answer in those units. Okay. So we've got 58.44 grams in every mole. There are 2 moles per liter. That's the definition of 2 molar, right? 2 molar means 2 moles per liter. And there are 150 milliliters per solution, okay? So I'm going to actually be a stinker here and be like 0.150 liters per solution. And I'm going to actually change the units of my answer I'm trying to derive to grams sodium chloride per solution. And this is because I'm doing this sloppy, they call it envelope math. You do it on a piece of scratch paper. And I'm like, okay, the way I set that up is going to give me grams of salt that I want added that I need for my solution. That way I'm not doing grams per 150 milliliters, So you run that math, 58.44 times 2 times 0.150. Who's got an answer in front of them from the lab midterm? 17.532 grams, all right, per solution. So the key thing is keeping the units straight. There's a tricky piece here where I just wrote it as liters instead of milliliters. But if you wanted to convert, you could go, okay, well, 150 milliliters, all right, and I want that converted into liters. So I need liters in the numerator and milliliters in the denominator. There are 1,000 milliliters per liter. So that would get us to this point right there. Does that kind of make sense? Everything's dimensional analysis.
How much, I guess, I don't know if this is important or not, but how much, I guess, milliliters of water would you use?
So yeah, thank you so much. The full answer would be, I take 17.532 grams of sodium chloride and add it to enough water to make 150 milliliters of solution. Because you don't really know how much liquid this many grams of salt is going to displace. Does that make sense?
Yeah.
Solid. Yeah. And displacement rates are going to differ all the time. So since you're doing this practically, it's like just make the solution, like dissolve it, and then bring it up to volume, and then mix it up more. You know? Yeah. Additional questions? Did this make sense for folks? Solid. And you'll see, like, all the math here is, most of the math here is all dimensional analysis, you know. Next topic, yes?
Could we go over PCR steps and components?
Yeah, totally. PCR steps and components is the next topic of discussion. So in a PCR reaction, I'm just going to express something to you right now. I have a learning objective that we're accomplishing in this review session. It can feel like, oh, John's just giving us answers. And it'll feel that way when you hit your lab final and you're like, wait, this is just, John he answered the question. What I'm trying to impart to you is the importance of asking questions, right? If there's an opportunity to ask questions of a person who has the information, ask the questions, take note of the information, and then when you need it, it's right there, you know? It seems very obvious, but sometimes it'll feel like it's easy to discount this as like, well, the class is useless. We just got the information told to us at the end. You know what I'm saying? Anyway, the point here, the learning objective for today is the value of asking questions, all right? And not being shy about, you know, asking them. And you'll encounter some people who aren't as helpful. And you'll encounter some people who are helpful. And I hope that you encounter mostly people who are helpful in your life. All right? Yeah. You're just stretching, but I just imitated you. I don't know why. Rude is what that was. PCR components. What do we need in a PCR reaction? Let's do a little bit of it together. Throw out something that we need in a PCR tube. All right? Just anything. That'd be great, yeah. We call it template DNA. The DNA that's going to be amplified, right? The template DNA. Anybody got something else that you'd put in that PCR tube? Buffer. Absolutely. Buffer is going to maintain ideal conditions, all right, for the reaction, okay? How about some water in there? How about some water, Scarecrow, right? Why not?
I can't remember the name, but what's the one thing that target the target site, you know?
Yeah, those are the primers. Very good. Primers, right? They target the region of interest, all right? And it's important to know, right? If you were running a PCR reaction looking for different things in the same individual, usually all you have to really swap out are the primers, because everything else, you know, is constant. So, like I'm saying, what I'm trying to say is, hey, if you were looking for markers that indicate you might develop cystic fibrosis or that you might develop Alzheimer's disease, you use the same template DNA, the same buffer, the same water, the same DNA polymerase, all right, all that stuff, but you use different primers to target different regions of the genome. Remember, this is specifically Taq polymerase or another thermostable polymerase, because your DNA polymerase would denature at the high temperatures that cause the DNA to separate. There's one last thing here. What's that? Oh, good point. Yeah, we do need magnesium chloride, because that provides the cofactor for DNA polymerase. Now, there's one more thing. Nucleotides. Free nucleotides because those are the building blocks that are going to be assembled by the DNA polymerase. Solid? The steps of the PCR reaction: hot, cold, middle, and then repeat, right? High temperature, low temperature, middle temperature. Usually around 95 degrees Celsius is the high temperature, okay? The high temperature denatures. The low temperature anneals. The middle temperature extends. So separate the double-stranded to a single strands, anneal the primers, all right? And then create the ideal temperature for Taq polymerase to extend the new strand of DNA. And then you repeat. And those are the steps. And it's high temperature, low temperature, middle temperature. And the specific temperatures, if you need them for your lab final, are in the protocol. Solid? Do you feel satisfied with the answer to your question?
I'm still trying to shake down the anneals and extends, just like the
100%. Yeah. Do you just need the spelling better or the explanation of it better?
the explanation.
You got it. All right. So here's the template DNA. It's double-stranded. The denaturation step opens up that DNA just by heating it up into two single-stranded structures. The annealing step is when the RNA primers attach. All right, right here. That's going to provide a 3' hydroxyl group so that the Taq polymerase can attach. And yeah, they're not necessarily RNA primers in PCR. I'm just going to say primers, okay, to make our lives a little easier. Don't worry about that statement that I just made, but I don't like to misspeak and then not clear it up. Anyway, and then the extension step is when the polymerase assembles the new strand, reading the template DNA as a guide, attaching the complementary bases. So annealing is when the primers attach, and extension is when the new strand gets built.
Thank you.
Awesome. Further questions about this topic? Let's move on to the next topic. Who's got another topic they'd like to discuss? Yes?
I admittedly missed the potato lab day, and you mentioned that specifically in terms of what you want us to know about enzymes.
Yeah. So you'll be in luck if you got the enzyme lecture, okay? The lecture portion, because it's the same enzyme topics of and what's an enzyme, what's a substrate, all right, what's a product, how do enzymes work, yeah, I've got 2 enzyme questions, and you could basically you just need to understand. I'm trying to not give you just the answer perfectly here. They're both really clearly in the protocol, and they're more about general enzyme things. So not specifically the potato enzyme lab. I know I'm repeating myself now when I say you got the same information in the lecture that you'll need for this, for the questions on your lab final. Sometimes there's overlap. Was that satisfactory? I can give you more detail.
You can't just give us the answers.
I'm going to try not to, but I'll be honest, that PCR one, I basically did give you the answers for the PCR question there, but that's okay. Yeah. I'll tell you, those enzyme ones are multiple choice questions. They're, yeah, awesome. And thank you for your understanding. Moving on, who's got another topic they would like me to provide clarification? We can always redo a problem you missed on the earlier lab midterm. Yeah. Number 12. Which one is that? Oh, sick. Yeah. I've got one right I've got some lab quizzes in front of me. Oh, well, I'll tell you. Let me see if you have to do that for the.
Yeah, because that's the one with the protocol was like.
Yeah. Okay. So I'm going to tell you that you don't have to do a problem like that one on the lab final. So I would say let's not worry about it. But there are questions about, hey, you did a dilution and you measured the absorbance as this much, and then use a standard curve to generate the diluted concentration, and then calculate the concentration before dilution. Does that make sense?
Yeah.
And how do you feel about those sorts of questions? You feel cool about that? Okay. Sick. Awesome. Okay, cool. Well, then in that case, yeah, that's another one where it's like, that's a fun, problem to do, but it's fun for me to do when there's no stakes, and I've done it a million times before. It's not fun to do when it's for a lab midterm. I don't mean to minimize how stressful these exams are at all. Okay. Additional, yes?
Is that what you were referring to for the bullet point absorbance spectra and color, or is that?
That is about pigments. Absorbance spectra and color is about pigments. So if you remember, the absorbance spectra was the thing where you got wavelengths.
Oh, and
Right.
that's the different. Yeah. Okay.
Yep. And you have all the different pigments on it, and they go like this and this and this. You know what I mean?
Okay.
Yeah. And then just remember, right, the wavelengths of light that it absorbs, those are the wavelengths that it does not appear to be. Yeah. The wavelengths that it does not absorb, that's the color of the pigment that we see, because that's the light that gets reflected or transmitted. Awesome. Yes, at the back.
So I was stretching.
You were stretching. I'm sorry, that's minus 10 points. You have a question?
I wanted to. Well, I was curious if you have like general forms of the equations for Hardy-Weinberg and chi-square, and/or did you say that just understanding the Newton equation is like enough math to get in?
If you can run, bless you, if you can run through the Newt question on the worksheet, then you're good to go. I don't even make you do the tricky part of like, determine p squared and then take the square root to get, or determine q squared, take the square root to get q, subtract p from it, and then, or subtract that from 1 to get p, and then use that to get the expected genotype frequencies. I don't even make you do that. Instead, I try to keep it more simple with giving you, I give you genotype frequencies. So it makes it, as long as you know how all the numbers play together, it makes it pretty straightforward. I try to make it straightforward at least, because it's such a complicated thing. So I'll give you, in one of these questions, I give you the genotype frequencies. And I ask you to, say, predict the number of individuals and determine the allele frequencies. And in the other question, I give you counts of genotypes. There are this many homozygous dominant, this many heterozygous, this many homozygous recessive. And then I just ask you, hey, is that population in Hardy-Weinberg equilibrium for that allele? Given those genotype counts. So that's a number of individuals. So you gotta be able to convert from genotype number of individuals of a given genotype to genotype frequencies, to allele frequencies, to genotype frequencies. You gotta know how to move between those numbers pretty easily. But what I'll tell most students at the start of this lab final is, if you're shaky at all on Hardy-Weinberg, save those for the end. Do the rest of the lab final, then go take a little break, have a snack, drink some water, like go up and down the stairs a couple times, like get yourself totally recentered, and then just spend 30 minutes probably doing the Hardy-Weinberg questions. Yeah, solid. Any other questions? Okay. Solid. All right. Well, what did that take? 45 minutes of recorded tutorial stuff and prep? That's great. That's going to end the recording right now. Thank you for watching.